The ultimate goal of protein engineering is to design proteins to carry out predicted function. However, we do not yet completely understand the rules governing protein folding and molecular recognitions. That makes design of proteins difficult. Therefore, protein engineers have invented some practical technique called combinatorial method, or called in vitro or directed evolution techniques. This technique based on two procedures. First, you make a pool of different amino acid sequences, usually produced by random mutagenesis applied to the corresponding genes, and then you select the appropriate structural functions among those pool. In selection, there are two approaches, in vivo and in vitro. So in vivo method, you use live cells. So just try to select the whole cells, which just contains the appropriate the target genes inside. In vitro, you just select the target genes specifically. The typical example for second one is the fuzzy display. You might understand better with fuzzy display. Here, we're going to see some techniques to produce random mutagenesis. In A, oligonucleotide can be synthesized to contain mixture of nucleotide at two codons. In red, in two regions, the corresponding amino acid will be 20 different types. So in total, 20 by 20 giving 400 different types will be produced. At the bottom, you can see some limited number of residues in this codon, mostly hydrophilic residues like glutamic acid, lysine, glutamine, aspartic acid, asparagine, histidine. Oligonucleotide library made this way, then now incorporated into the gene of interest. B is called error-prone polymerase chain reaction. In this case, PCR technique based on the DNA polymerase is designed to produce the less fidelity, so that some mutation is shown in something like that during the replication occur, so that the final product will be the certain random mutagenesis shown in red color. C just indicate a technique called DNA shuffling. A gene is first cut into pieces and then regenerated using a DNA polymerase. Polymerase introduces mutations similar to error-prone PCR. Additionally, the pieces of DNA get mixed so that the mutations from separate copies of the gene can be combined. Fuzzy display was made in 1985 by George Smith in University of Missouri. Fuzzy display typically utilizes bacteriophage M13. This filamentous phage contains single-stranded DNA encased in a protein coat. There are 5 types of coat proteins, including major G8P and G3P. At the end, we can construct fusion protein of phage coat protein plus the target peptide or protein you want to produce by genetic engineering. And then you just prepare the phage particle shown like this. Here you can see G3P at the end, usually 5 copies, but one of them has a fusion protein, red one. Or you could construct the GAP protein below. But in this case, you do have some multiple number of fusion proteins. So the top one is called monovalent, and then the lower, the multivalent. Anyway, not shown in this picture, but the corresponding gene for the fusion is encoded, encapsulated inside this particle. Fuzzy display experiment typically includes 3 steps. First, a library containing around 10 to the 8th magnitude of phage is screened to select the phage that contains the fusion protein that you want. And then you just proliferate the number of phage, selected phage in bacteria, and then characterize, including the sequencing the genes. In this picture you see this kind of library is used to select, in this case, some the immobilized antibody. So you wanna select antigen which has a binding capacity to this specific antigen. So after you pour of your phage and then wash, the unbound form may be washed away. Only tight binding phage could be selected, and then you could characterize. In the section below, we're going to look at some application of phage display. Blood coagulation involves trypsin-like serine proteinases, including plasmin, kallikrein, factor 9A, and TFF7A. While there would be an important clinical benefit to engineering specific protease inhibitors, the active sites of these enzymes are highly conserved, showing SIS 81% identity, making the design of specific inhibitors difficult. Kunit domains make up one family of protein inhibitors of trypsin-like proteinases. Kunitz domains, you can see APPI, LACIDI as an example, having about 60 residues, 3 disulfide bonds for stabilization, and highly conserved for their structure. Each Kunitz domain could bind to one or more proteinases. Affinity and specificity of Kunitz domain to tyrosine-like proteases are determined by two loops. You can see two protein structures of APPI in A and LACID1 in B, both as primary binding site in green color, raised to 11 to 19, where P1 is highly conserved for lysine or arginine, and then this region binds to the proteases subsite labeled S5, 4, 3, 1, 1 prime, 2 prime, 3 prime, 4 prime. And second loop raised to number 34 to 39 in orange color in both binds to the proteases or just to contribute to making the correct structure of binding loop. Here you can see the application of Fuzzy display on Kunitz domain. Two groups started with different Kunitz domain. One, LAC1D1 by Protein Genetic Company in United States, and second, APP1 by Genentech. And then they end up with mutagenized protein that has a high affinity to the different Kunitz, that is kallikrein. So you can see the sequence of the starting the Kunitz LAC1D1, as you see here in red box. After application of Fuzzy display, they've got the blue box, which has a very high affinity to kallikrein. Okay, Ki value is 40 picomole. That could be defined as something like this. So dissociation constant between protein and this inhibitor. When you compare two results, the fact that these two Fuzzy display libraries arrived at very similar sequences, having started from different scaffold and library designs, can be described as convergent in vitro evolution. Next experiment was competitive or subtractive sorting to select inhibitors specific to 7A in this case. To achieve this purpose, they added TF9A into the Fuzzy pool to remove the Fuzzy that has a high affinity to the 9A. Then they select against 7A. The result in blue box with nanomolar affinity. Although this shows affinity to factor 9A and calcitine to the similar way, the substrate sorting thus shifted in vitro selection towards mutations that were tolerated only by the desired targets. How do the mutations obtained by the Fuzzy display improve binding affinity? Model structure suggests maximizing the complementarity between two components.